Hi everyone, in this video I'll be going through the topical exercise for transport in plants. Let's begin with the first question. In this question, it provided you two diagrams, one of the phloem and one of the xylem. And then I have to admit there isn't really a question here, but they want you to, in a sense, identify what's 1, 2, and 3. Okay, so what exactly is all these things you're seeing? First and foremost, when you look at this over here, these are the sieve elements of the phloem vessels. Okay, here you will see the companion cells. How do I tell these are companion cells? That's because you still see that they have all the necessary parts of a cell, from the vacuole to the nucleus. This is very important because the companion cell supplies nutrients to the sieve element cells, keeps them alive. On the right, you see that there are xylem vessels. These are tracheids. Okay, how do I know? Because they are thinner and more narrow and long, whereas these one are the these are the vessel elements. Okay, so what are their functions? Okay, so the answer is B. Number one are the sieve elements. That's what they transport amino acids. They can also actually transport sucrose, so this is actually still okay. Next, number 2, the companion cells. They supply energy to the surrounding cells, more in the form of nutrients and ATP. Okay, so these two options are okay. Next, number 3, that is the vessel element. So it transports water as well as mineral salts. So the best answer is B. Next, let's go to number 2. Diagram 1 shows the longitudinal section of a plant stem, and diagram 2 shows the transverse section. They ask you which part on diagram 2 contains the components shown in diagram 1. Okay, so what in the world is diagram 1? Diagram 1 is again showing you the phloem filled with, which includes the companion cells as well as the sieve elements. Okay, you can even see the sieve plates over here from a side view. So where will you find the phloem? The phloem are often the vessels that are on the outside. Okay, the xylem are the ones often on the inside. So let's try to make sense of why you will find the phloem on the outside and the xylem on the inside. The best explanation I have is that when we look at the xylem vessels have the additional secondary cell wall. So they have a lot of lignin. This gives structural support. So in a sense, I think that the xylem vessels are found on the inner side because they help provide structural support. You want to provide a structural core to the plant to keep them upright. Right, so that's what I think is the reason why xylem vessels are found on the inside, whereas phloem are found on the outside. Okay, so what's the answer? Answer is A. Okay, phloem is found on the outside. Next, which process contributes most to the rise of water in the xylem? The answer is D, transpiration pool. Okay, the others are also important, except that transpiration pool is the most important. Next, 4, which combination of factors will cause a decrease in transpiration rate in plant? Transpiration rate is heavily tied to the idea of evaporation of water from the leaves. So anything that will increase evaporation rate, so increase transpiration. Anything that decreases it will also decrease transpiration. And so the combination that will result in a decrease in evaporation rates will be if there's less air movement, if there is increased humidity, if there is decreased light intensity. Okay, because light intensity can also affect whether there is going to be evaporation, and finally temperature. Okay, decreased temperature too. So actually C would be the answer. Next, we go to question 5. Diagram shows sections through a leaf and a stem. Where does translocation takes place? Translocation is related to the phloem. So the phloem translocates sugars such as sucrose. Right, as mentioned earlier, you'll find the phloem usually on the outside. So this is the phloem, right? Then what about in the leaves? So how I make sense of it is this way. Okay, if I look at the diagram to the right, you find that this is what you see. These two diagrams are the same. Phloem is on the outside, xylem is on the inside. In a sense, as the plant grows, as the plant grows up, right, and then after that it starts to branch out, and when it goes into the leaves, it makes sense that the phloem, which used to be on the outside over here, ends up at the bottom. So this is where the phloem is, right? The phloem is on the outside at the moment. You find that the phloem ends up at the bottom now. Okay, whereas the xylem, which was once at the inner side, right, when it ends up in a leaf, now it ends up on top. Alright, so that's how I relate to why do you find the phloem on the bottom when you look at the leaf, whereas in the stem, the phloem is found on the outside. Okay, so the answer is D. Next, let's look at question 6, structured question. Here, they show you an experiment where they have 3 pots. Each with an equal volume of water was given to each plant. Plastic bags were placed around each pot of D and F. E was enclosed totally with a plastic bag. Each plant was placed in a sunny position by a window. The loss in mass of each plant was calculated each day for a period of 10 days. So they ask you to describe a procedure that should be followed to enable the loss of mass each day to be calculated. So logically, what you want to measure is measure the mass at the start of the day and at the end of the day. So this is the procedure. Number 1, you want to weigh the plant without the plastic bag after water was given at the start of each day. Okay, and then after that you want to weigh the plant without plastic bag again at the end of each day. So why is it important that you weigh the plant without the plastic bag? Well, because if you compare between D, E and F, actually the amount of plastic bag used is quite different. D and F, quite similar. But when you look at E, a lot more plastic is used. So if you were to just weigh each pot, you would find that E may start off with a little bit more weight, a little bit more mass because there's more plastic bag. So the logical way would be to remove all the plastic bags, then you weigh them, because you start off with the same pot, same amount of soil, same amount of water. Okay, so in order for you to calculate the loss of mass each day, you take B minus A. Then you can find out the loss of mass. And this loss of mass, actually, that mass that is lost is water being lost. That's the real thing that's being lost from the plant. Next, let's go to question 6b. Name the process responsible for any loss in mass by the plants. And the answer is transpiration. They only ask you to name it, so you just have to write transpiration. Next, suggest and explain which plant would lose the most mass over the 10-day period. If you look at this over here, you'll find that when you look at D, E, and F, some things are different. Number 1, when you look at D, there are more leaves compared to in F, right? So if we just compare D and F, you expect that more transpiration will take place in D because there are more leaves and more surface area for transpiration to take place. You compare between D and E, the whole plant is enveloped in plastic bag. That means to say within this plastic bag region, there's very high humidity. If there's high humidity, it will result in low transpiration rates because at the end of the day, transpiration is affected by how much evaporation takes place. So as a result, if you compare between D and E, D will still be the winner. There will be more transpiration taking place. Okay, so plant D, why? Because there's more leaves and hence a greater surface area for transpiration to take place. Last, when we look at D, suggest why the pots plant D and F were enclosed in plastic bags. Well, because we want to reduce the amount of water loss from direct evaporation from the soil. If it directly evaporates from the soil, then we won't be able to tell that transpiration has taken place. Next, let's go to question 7. In the investigation, plants were provided with carbon dioxide that contained radioactive carbon. This allows scientists to track the movement of carbon in the plant. So here is that container where they have the radioactive carbon dioxide. So two groups of aphids, A and B, were placed on a stem, and one near the top and one 65 cm below. So there are aphids in this region. Aphids are insects that suck on the sap of phloem. And then you also have aphids over here, 65 cm away. One at the top, one at the bottom. So the first question, they say after several hours, the aphids were removed from the stem and tested. They were found to contain radioactive carbon. State the name of substance that contained the radioactive carbon which was taken in by aphids. The substance would be sucrose. Okay, sucrose is a sugar, and this sugar was made from, in part, glucose. Okay, you cannot say that the substance is glucose because glucose is not the thing that's transported through the phloem. It is sucrose. However, sucrose is made from glucose, and this glucose came from the Kelvin cycle. And how was glucose made in the Kelvin cycle? Well, it used the carbon dioxide in the air, and because the carbon dioxide in this case is radioactive, this glucose became radioactive too. And when used to make sucrose, the sucrose in turn became radioactive. Next, B. State the name and plant tissue where the substance containing the radioactive carbon would be found. The answer is the phloem. The phloem is the one that transports or translocates sucrose, and that's where the aphids were sucking from. The time taken for the substance containing radioactive carbon to travel from B to A was 2.5 hours. So from B to A, that's the time it took, 2.5 hours for the sugars to be transported down. So they ask you the rate of movement. It's not exactly a biology thing. We just wanted to see if you can make sense of the data given to you and learn how to analyze it. So the rate of movement will go something along lines of 65 cm. That's the distance traveled over 2.5 hours, and this is the answer you can get. Okay, per hour. So that's the movement per hour. Okay, last free response questions. They ask you to describe the similarities and differences in the function of the xylem tissue and the phloem tissue. So some similarities include that both transport substances throughout the plant. Second, both transport substances, they are dissolved in water. So whether it is mineral salts in the xylem or it is the sugars like sucrose in the phloem, both of them have to be dissolved in water for transportation. So those are similarities. What about the differences? When we look at the differences, xylem transport mainly water and dissolved minerals, whereas the phloem transport mainly sucrose dissolved in water. So when we look at the functions, it is the difference here is what do they transport. Next, difference number 2 dives into more of where they transport from and to. The xylem transports substances from the roots to the leaves, while the phloem transports substances from source organs to sink organs. Example of source organs include things like the leaves. Example of sink organs includes things like fruits. Okay, so this is the where they are transported. Next, when we compare the xylem and the phloem, we know that the xylem has a lot of lignin in their secondary cell wall. And so another function of xylem is that they can provide support to the plant, structural support. Because the phloem lacks lignified cell walls in their therefore, they cannot provide structural support. So that's another comparison of function. Next, when we compare the movement of substances in the xylem, into the xylem, we know that the movement of water into the xylem is a passive process of osmosis, while sucrose movement into the phloem is driven by active transport. So cells that are surrounding the phloem actually actively pump the sucrose into the phloem via active transport. Whereas primarily a lot of the water moving into the xylem move in via passive processes like root pressure or like transpiration pull, capillary actions. All these all help in moving the water into the xylem. Last difference, movement in xylem into the xylem is driven largely by transpiration pull, while movement in the phloem is driven by bulk flow. Okay, another word you could use other than bulk flow is pressure flow. Okay, and we learned that what pressure flow is the idea that there are sugars being pumped into the phloem, and because of the difference in the amount of sugars between the source and sinks, this will help in water flowing as a result. Okay, last, when we look at the last question, they ask you to describe the process of transpiration. So if you ever asked to describe the process of transpiration, this is the depth that you need to go to. Number one, you want to define what transpiration is. It is the loss of water as water vapor from aerial parts of the plant. Aerial parts means parts that are above ground. Okay, and they say loss of water as water vapor. Next, so where does this water come from? The water actually comes from the lining of water on the surface of mesophyll cells. This water evaporates from the surface of mesophyll cells to form the water vapor in the intercellular air spaces of the leaves. So what do I mean by that? And what are mesophyll cells? Mesophyll cells are these cells you find over here. Okay, if you look at the diagram below. These cells are mesophyll cells. If you recall from the lower GH, these are spongy mesophyll cells. These spongy mesophyll cells also come with a lot of intercellular air spaces. Okay, one thing special about the spongy mesophyll cells is that they have this lining of moisture on their surface. This is where the water will evaporate from into the intercellular airspace. Okay, and from there they would then leave the guard cells or the stomata into the environment, and that's what transpiration is. So point number 2 is saying that the water lining these spongy mesophyll cells will evaporate into the intercellular airspace. Where does this water vapor go? The water vapor then goes from the intercellular airspace out of the stomata, okay, into the surrounding spaces. That's point number 3. And this loss of water from the surface of the spongy mesophyll cells ends up pulling more water from surrounding cells. Okay? So as more water is being lost from the surface, then more water will be pulled from inside to replace any water that's been lost from the surface of the spongy mesophyll cells. And as more water is being pulled to the surface, then it will eventually be pulled from the xylem vessels. And that is what pulls water up the xylem vessels, or what we know of as transpiration pull. Okay, so that's all for this particular chapter. If you have any questions, please feel free to contact me. Thank you.